Hello and welcome back. This is our second video in our video series for Chapter 6, Expression of Biological Information. My name is Miss Delia, and in this video, we're going to go into 6.2, DNA Replication. If you're ready, let's get started. So let's have a look at the learning outcomes for this subtopic. By the end of this lesson, you should be able to explain the semi-conservative replication of DNA, the enzymes and the proteins involved in DNA replication, and last but not least, explain the mechanism of DNA replication and the enzymes involved. So we're going to start with DNA replication itself. Now, you probably already know what is the definition of DNA replication, but we'll just go through it bit by bit. So this first section is talking about what it is. DNA replication is the process of synthesizing new DNA molecules during the S phase in interphase of the cell division, in which the hereditary material is duplicated or become double. So, kita punya DNA akan membuat salinan kepada diri dia sendiri dan ini berlaku dalam S phase kalau kamu masih ingat kita punya cell cycle. Okay, so it will occur by semi-conservative replication, and it is a mechanism that is based on Michelson and Stahl's experiment. So these are two scientists who confirmed this theory. So this is the semi-conservative replication, which is saying that one strand of DNA will produce two strands of DNA that is half new, half old strand, and so on. So yeah, so this is the semi-conservative replication model. The original DNA will act as a template. Okay, original DNA itu yang kamu nampak warna biru gelap di bawah. Itu adalah yang original dan dia akan digunakan sebagai template ataupun sebagai asas. Okay, the original copy. And then dia akan buat salinan. It will produce two DNA molecules. Each will consist of one old and one new strand. So if you're looking down here, you see the old strand is in dark blue and the new strand is in light blue. So this is the semi-conservative replication model. Before this, people were debating whether it is a fully conservative replication model. Kalau fully conservative means yang lama dia terbuka, dia buat salinan, then dia buka balik, yang lama akan gabung dengan yang lama, yang baru gabung dengan yang baru. There is also another one that I'm not quite sure what the name of that model was. It was kind of like instead of a full strand, it will be half strand. A bit of this strand is old, a bit of this strand is new, a bit of this old, a bit of this new, a bit of this old. But basically, what you need to remember for your syllabus now is that our DNA replicates according to the semi-conservative replication model. Model-model lain kita tidak perlu hafal, tidak perlu baca untuk syllabus kamu. Okay, so this model was confirmed by these two, through the experiment by these two scientists. So Michelson and Stahl, which are the two gentlemen down here, they carried out this experiment. Saya akan baca lah step ataupun procedure yang diorang gunakan untuk eksperimen diorang. So this is the details of the experiment carried out by these two gentlemen. Okay, to know how DNA replicates themselves, they started by labeling the parental DNA with 15N. Okay, ini 15 adalah proton number of nitrogen, of a nitrogen atom. So I'm sure you remember from chemistry, you have your proton and nucleon number. Hold on, let me just be sure which number this is. All right, sorry, I made a mistake. The 15 is not the proton number, but it is the nucleon number. So itu ada 15 itu adalah nucleon ataupun atomic number untuk nitrogen. So the original strand was labeled with 15N by growing E. coli bacteria for many generations in a medium that contained 15NH4Cl as the sole nitrogen source. Maksudnya dalam satu medium tu, contoh macam agar-agar punya blok, okay, dia orang berikan banyak sumber zat, tapi untuk zat nitrogen, for nitrogen for these bacteria, they put it in the form of this chemical that uses nitrogen with atomic number 15. Selepas beberapa generasi bakteria tu hidup dalam medium tu, dia punya DNA akan menggunakan 15N. Okay, after you are sure that this bacteria only contains DNA that has 15N, it will be transferred to a medium that contains 14NH4Cl as sole nitrogen source. So sekarang dia bakteria E. coli ini, dipindahkan ke dalam agar-agar baru yang ada 14 nitrogen. 14 nitrogen punya atomic number. So one sample of bacteria was allowed to divide once to provide the first generation of cell. Another sample of cell was allowed to divide twice in order to obtain the second generation of cell. So diorang ada dua batch lah. First batch itu lepas satu generasi dia hasilkan first generation of bacteria. Diorang ambil sudah untuk buat analisis. The second batch kasih biar dulu, biar dia sampai second generation, baru diorang ambil dan analyze. That is what these points mean. DNA was extracted from these cells and their densities was determined by centrifugation in cesium chloride ion. What is meant at point number 5 is basically they take the DNA out of the cells, they extract it, and then the densities was determined through centrifugation, yang kasih pusing laju-laju, in cesium chloride ion, which is the medium used. If the DNA contains only 15N nitrogens, then they are denser or heavier compared to DNA that have 14N. So, DNA with 15N, the former, would form a band below the latter, which is DNA with 14N, in cesium ion solution, cesium chloride solution. This hybrid DNA that consists of one 14N and 15N strand would bend halfway between the 14N DNA band and 15N DNA band. So that what is being said there is being illustrated here. Okay, ayat-ayat tadi digambarkan di sini. Okay, so the original DNA consists of only 15N. So bila diorang analysis, they put in the centrifuge, it's going to be here in the test tube. So the parental strands will be heavy because they are all 15N. The first generation, it will produce this kind of DNA. So half of it will be 15N, the other half is 14N. So it is called the hybrid DNA. The position on the test tube will be in the middle here. In the second generation, after we allow this to grow, okay, for one more generation, it's going to produce 4 strands of DNA of the same copy. And then you will see that these are half 15, half 14N. These two in the middle are only 14N. So that's why when you centrifuge it, you're going to get the hybrid DNA, which is in the middle, and the new light DNA on top. And if you centrifuge it another time, okay, if you let it grow another time, you will have more layers that are very light, that are high, very light blue, and less layers that are hybrid or in the middle of the test tube. So inilah cara Michelson dengan Stoll tengok diorang punya eksperimen punya result. Kalau dia makin bawah means itu adalah DNA lama, makin atas DNA paling baru. Okay, so the result is that for the first generation of cells, the DNA band was precisely halfway between those of 14N and 15N DNA. With the second generation cells, 2 bands were now formed. Half of the DNA molecules were the hybrid type and the other half of pure 14N DNA. So the conclusion from this experiment is that Michelson and Stoll concluded that DNA replication will occur through the semi-conservative model. And when a double helix replicates, each of the daughter strands will have one old strand and one new strand. Kalau kamu lupa ayat-ayat yang penuh, kamu tengok sudah diagram biru di sebelah ni. Diagram biru ini sudah menggambarkan poin untuk semi-conservative model. Michelson and Stoll are just two scientists who prove this theory. You used to have to understand, memorize, and explain Michelson and Stoll's experiment. But with your current syllabus, you don't need to know about the experiment. Yes, sekadar saya beritahu kamu untuk kamu faham, then kamu boleh ingat yang perlu hafal adalah the part about semi-conservative model saja. Okay, I hope that is clear enough for you. Now we are going to go into DNA replication process itself. So it will occur during S phase. It will have one parental DNA produce one new DNA, and it involves three steps. The three steps are initiation, elongation, and termination. It will involve several enzymes and proteins. Okay, so ini adalah beberapa enzyme dengan protein yang terlibat. The first enzyme is DNA helicase, which functions to untwist and unwind parental strands at the parental DNA strands at the replication fork. You will have the SSBP, which is short for single-stranded binding protein. It binds to unpaired DNA strands, keeping them from rejoining. The third enzyme is DNA gyrase, which also has a different name called topoisomerase. Okay, the function is to break, swivel, and rejoin the parental DNA ahead of the replication fork. The fourth enzyme is primase. So this will synthesize RNA primer at the 5' end at the parental DNA and synthesize the beginning of each Okazaki fragment at the lagging strand. The fifth enzyme that we'll look at is DNA polymerase 3. Okay, memang nombor 3 dulu, DNA polymerase 3 dulu. It will catalyze synthesis of new DNA by adding DNA nucleotides complementary to the template strand from 5' to 3' direction. And then you have DNA polymerase 1. It removes the primers in DNA strands and replaces them with DNA nucleotides. And last but not least, you have the DNA ligase, which joins DNA fragments together by catalyzing the formation of phosphodiester bond. Okay, ini adalah senarai enzyme dengan function dia sahaja. So this is just a list. It's easier to understand how they function and how they work when we're looking at videos and when we're looking at each step of DNA replication. So don't worry if you don't understand these stuff yet. We will finish 6.2 and then you can come back to this at your own time and try to understand each one. What I would like to say is once you remember the enzymes and the proteins involved, it is very easy to remember the steps of DNA replication. So kalau kamu boleh ingat enzyme dengan protein ini, sangat mudah untuk kamu ingat step-nya. Okay, so this is the mechanism of DNA replication. This is one of the diagrams that show from step 1 until step 7. Okay, so daripada mulanya penghasilan replication bubble sampailah habis DNA replication, boleh didapatkan dalam diagram seperti ini. Okay, so just an overview. Before I go into the step-by-step explanations, I would like to tell you what is a replication fork or a replication bubble. So replication ini, dia akan mula dengan membuka DNA. Tempat yang DNA dibuka itu, dia akan mula pada satu titik, okay, contoh pada titik ini, dan dia akan membesar atau proses ini akan berterusan ke kiri dan kanan. Once you start replication, it will begin at an origin of replication and go left and right. So here it is marked that this is the origin of replication. And where you see the DNA unzipping or unwinding, that is called the replication fork. This circle thing is called the replication bubble. Kawasan yang sudah dibuka dipanggil replication bubble. Tempat di mana DNA sedang dibuka, macam zip sedang ditarik, adalah replication fork. Okay, so this is replication fork. We can divide DNA replication into 3 stages, as I've said before, which is initiation, elongation, and termination. So if you're ready, let's get started on the steps of DNA replication. Okay, first step is initiation. Untuk mulakan DNA replication, kita mula dengan step pertama, initiation, where we will use the first enzyme. The first enzyme is DNA helicase. DNA helicase ini dia macam gunting. It's like scissors or it's like the thing you pull on your zipper. Okay, dia adalah tukang buka DNA. So the first point is DNA helicase unwinds the double helix of DNA by breaking the hydrogen bonds between the two strands at the origin of replication. Origin of replication is the starting point for DNA replication. So enzyme itu akan cari starting point itu ataupun origin of replication dan dia akan mula buka DNA. Dia buka DNA dengan memutuskan hydrogen bond di antara base. So you will have a replication bubble produced. In prokaryotes, in bacteria such as E. coli, there is only one origin of replication. And since bacterial DNA is circular, it will look like this diagram. Pemandangan DNA bacteria adalah bulat, circular, dia akan nampak macam diagram sebelah kiri ini. If it is in a eukaryotic cell, eukaryotic DNA is linear. Okay, lurus saja DNA untuk eukaryotes dan jauh lebih panjang, lebih besar. So akan ada lebih daripada satu origin of replication pada setiap kromosom. Okay, so on each strand of DNA, one chromosome will have several origins of replication. DNA helicase will go and find each of the origins of replication and start to unwind the DNA. So sini dia unwind satu bubble, satu bubble, satu bubble. Dan setiap bubble ini akan membesar sampailah bubble-bubble itu gabung dan akan pisahkan dua parental strand tadi. Dia akan jadi dua daughter DNA molecule. And these are images taken under the microscope to show what they look like during replication. Okay, we're going into point number 3. The separated DNA strands are stabilized by single-strand binding proteins. Okay, kita sudah pergi kepada protein ataupun enzim kedua. DNA ini kalau kita biarkan dia akan gatal. Dia mau juga pegang tangan. Dia mau juga gabung balik dengan kawan dia tadi. Jadi, untuk mengelakkan DNA yang sudah dibuka ini untuk gabung balik, kita gunakan SSBP ataupun single strand binding protein untuk pegang dia supaya dia tidak tergabung balik. So, as you can see here, all these circles are single strand binding protein. The thing about winding and unwinding things is the end of the line will get coiled. Kalau kamu boleh cuba ambil benang kah, ribbon kah, ataupun penghujung mask kamu. Okay? I'm gonna zoom in on my camera. I'm going to show what is happening in this diagram over here. So say I have, this is my mask, I'm just borrowing it for a bit. Okay, so contoh ini adalah DNA kita yang sudah coil. Okay, if I am unwinding here, kalau saya buka di sebelah sini, yang sebelah sini, ahead of the replication bubble, dia akan makin terikat, makin tersimpul. So you see here, before I do anything, dia macam longgar kan? Bila saya unwind saja di sini makin ketat. Dan ada kemungkinan dia akan tersimpul. So this can happen in our DNA. Your DNA is quite long, and if you just unwind and you don't do anything at the other end, DNA kamu akan terlilit. Your DNA will become coiled, and then that you cannot continue with anything else after that. So we use this enzyme called DNA gyrase or topoisomerase. Itu adalah dua nama untuk protein yang sama, enzyme yang sama. So function dia adalah untuk mengelakkan DNA kita terbelit dan terlilit di bahagian sini, dia akan putuskan DNA, kasih pusing, dan sambung balik. DNA gyrase will help relieve the strain on DNA by breaking, swiveling, and rejoining the DNA strands. How you want to remember is it just prevents supercoiling of DNA. Dia mengelakkan DNA kita terlilit. Continuing with initiation, both strands of parental DNA will now become templates for the new complementary DNA strands. Dua-dua ini akan digunakan sebagai original copy. Kita akan membuat salinan berdasarkan template ini. DNA primase, which is the next enzyme, will catalyze the formation of RNA primer at the 3' end of the template. So, kita boleh tengok di sini, we have your RNA primase, is not shown here. Okay, di sini dia tiada tulis RNA primase. Tapi kalau kita tengok diagram ini, direction of replication is from right to left. Okay, direction of replication is from here to here. So the DNA primase will start by putting an RNA primer. Sepatutnya dia gunakan DNA nucleotide. Tapi enzim-enzim dalam badan ini kadang-kadang diorang kurang yakin ataupun dia tidak dapat cari DNA. Jadi untuk membantu enzim lain buat kerja, DNA primase akan letak satu RNA primer di mana-mana tempat yang dirasa sesuai dan RNA primer ini akan diganti nanti. Okay. So after the RNA primer is placed by DNA primase, we are going to go into step number 2. The second step is elongation, pemanjangan DNA. DNA polymerase 3, mesti mula dengan number 3 dulu ya, will bind to the primer and add 3 DNA nucleotides to the 3' end of RNA primer complementary to the template. So tadi DNA primase letak satu primer, RNA primer. DNA polymerase 3 datang cari primer itu dan dia akan tambah DNA nucleotide yang betul. Macam mana dia decide mana satu DNA nucleotide? Dia tengok berdasarkan template. Tambah. Mula daripada 5' end, memanjang ke arah 3' end. So, elongation will occur from 5' to 3' direction of new strand. Continuing with elongation, both these new strands are synthesized in 5' to 3' end, and the daughter strands are called leading strand and lagging strand. Okay, leading strand, dia satu saja RNA primer dan dia akan terus buat satu DNA yang panjang-panjang. Lagging strand pula, dia akan ada beberapa RNA primase dan nanti perlu disambungkan semula. The leading strand will elongate continuously in 5' to 3' direction towards the replication fork using only one RNA primer, which is what I mentioned just now. The lagging strand will elongate discontinuously away from the replication fork, and they will form Okazaki fragments using several RNA primers. Kenapa ada satu leading, ada satu lagging strand? Sebab semasa replication fork, semasa DNA itu dibuka, satu bergerak ngam-ngam di belakang DNA helicase. Satu lagi bergerak bertentangan arah dengan DNA helicase. So, dia tunggu DNA helicase sudah jalan sedikit, baru dia boleh tambah satu primer. Tunggu DNA helicase jalan lagi, baru dia tambah satu primer. Itulah dia terputus-putus. Later, you'll watch the video and you will see this being illustrated. So, those fragments ataupun cebisan-cebisan itu dipanggil sebagai Okazaki fragments. Untuk Okazaki fragment pada lagging strand itu akan digabungkan juga nanti, jangan risau. So, we're going to continue with the steps. Now, step number 12, or point number 12, DNA polymerase 1 will replace the primers with DNA nucleotide before the DNA ligase can join the fragments together. So sekarang DNA polymerase 1 datang cari yang primer RNA, dia gantikan dengan DNA nucleotide. Lepas tu DNA ligase boleh sambungkan mana-mana cebisan yang tidak tergabung. Okay, DNA ligase will come and join the Okazaki fragments into a continuous DNA strand. When will this DNA replication end? It will end at termination, which is when replication complete. Proses ini akan berterusan sehingga seluruh DNA itu sudah siap disalin. It will continue until replication completes, where you will have two identical copies of the original DNA bond. Each DNA molecule will now have one parent strand and one new complementary strand, as according to the semi-conservative model. Okay, so just a quick summary. In this subtopic, you should be able to explain semi-conservative replication of DNA. You should be able to explain the enzymes and the proteins involved in DNA replication. So kamu perlu pandai list dan nyatakan functionnya. And also explain the mechanism of DNA replication and the enzyme involved according to this diagram. And I would recommend that you follow the steps that we have given you in this lecture note. So akan ada initiation, elongation, termination, dan ikutlah poin-poin yang diberikan dalam lecture note ini. Sebab itu yang paling mudah untuk kamu faham. Okay, so that was it for the short summary. Now we'll have a quick look at the video lesson on DNA replication. So we're going to watch this video and you're going to try to identify the steps that are involved and also try to remember which protein or which enzyme it is and what they are doing in this video. Okay, so we'll begin going into the nucleus. Now we are looking at a DNA, and this is a DNA helicase enzyme. So it's going to find, as you see here, it found the origin of replication, and it's going to unwind the DNA by breaking the hydrogen bonds. So as it unwinds the DNA, it is creating the replication fork. Okay, kita tengok ini cara DNA helicase memotong dan membuka DNA. Okay, so it's going to progress in this direction. Now you see here, these are single-stranded binding proteins ataupun SSBP. They are going to bind to the separated strands of DNA so that they do not come back together, okay, so that replication can continue. Okay, so we see here there will be many SSBPs and they're going to bind to the DNA. Here we have new DNA being formed. When the new DNA is from the direction of origin of replication towards replication fork, ataupun dalam erti kata lain, DNA baru itu sedang ikut ngam-ngam di belakang helicase, itu adalah leading strand. So as you can see here, this is the leading strand. It's following right behind DNA helicase. What is elongating it is DNA polymerase 3 from 5' to 3' direction. So it is a continuous replication of the leading strand. Down here is the lagging strand template, also formed in 5' to 3'. And they are discontinuous. So kita nampak sini dia punya DNA, ada bahagian yang tidak terjalin, ada bahagian yang not connected. So when it is not connected, it is the lagging strand, and the lagging strand consists of Okazaki fragments. Setiap cebisan yang tidak bersambung ini digelar sebagai satu Okazaki fragment. Okay, and we see here this green one is RNA primase. Kadang-kadang RNA primase, kadang-kadang DNA primase, kamu tulis sejarah primase. Dia akan tambah satu hijau itu adalah RNA primer. Dia tambah sedikit saja. Dia ikut ngam-ngam di belakang helicase. Dan DNA polymerase 3 akan panjangkan DNA dengan menambah DNA nucleotide baru. DNA polymerase 3 will find the RNA primer and then elongate the new strand by adding new DNA nucleotides that are complementary to the template. And then we will have polymerase 1. DNA polymerase 1 comes here and replaces the RNA. DNA ligase comes and joins the fragments together by catalyzing formation of phosphodiester bond. So you can see here you have the leading strand, the lagging strand, the template strand. You can see several of the enzymes that are working together to make sure that this replication can continue. And you have the replicated DNA. Replicated DNA, once the hydrogen bonds are formed, they will eventually form the double helix again. Lepas ini dua strands na bergabung, dia akan automatic mau belilit jadi double stranded. Okay? So that is the replicated DNA. So yeah, this is a video showing how DNA replication works. If you are using the PowerPoint version of these slides, you should be able to play this video on PowerPoint. If you're using the PDF, unfortunately, you're not able to play the video. Okay, so we have finished this part of the chapter. What should you do now? Now I would ask you to please make your own notes for DNA replication and answer the Google form quiz that is linked in the GC. That's all for me. Thank you very much for your time and attention, and I will see you again in the next video. Bye!